Hi everyone. Today's lecture is dedicated to action, or to use a more technical term, to motor control. This is the last lecture before midterm 2, so we're going to cover into as much detail as we can over the span of 2 hours this topic. So to briefly go over the lecture objectives, we will look into understanding how motor signals can cause muscle contractions that yield a desired movement. We'll try to describe the role of so-called central patterns generators in conducting complex stereotypical voluntary movement, such as walking. We will look at the form the motor impairment might take as a result of cortical damage or spinal cord damage. We will try to examine how M1, that is motor cortex, and also parietal cortex neurons code the direction of movement and the endpoint of movement, respectively. We will look at the complementary roles that the premotor cortex and the supplementary motor area play in coding information about movement. And we will also discuss how the cerebellum is capable of refining movement patterns and coordinating to exquisite detail aspects of movement as related to gait, to posture, or even to speech. And we will see exactly what cerebellar dysfunction might look like as a result of lesions to the cerebellum. With regard to the practical part of the lecture, today we will conclude with a discussion of the methods part of an empirical paper. If we talk about the functional architecture of the motor system, it should come as no surprise that we are talking about a very complex system that relies on many regions and very different pathways for feed-forward and feedback transfer of information. However, one distinction that I would like to draw your attention towards is that between planning or programming movement and execution of movement. Now, the regions that facilitate both planning and execution are quite a few, and we will not have the opportunity to talk in detail about every single one of them. However, I will draw your attention to just a number of them, which are shown here with the magenta colors. But before we move into a more detailed discussion of these regions, we should probably tackle a much simpler question, yet a fundamental one. How is movement generated? And to answer that question, one of the most useful instruments is EMG, or electromyography. In the same manner in which EEG is used for the purpose of measuring electric potentials on the scalp as generated by the brain, we use EMG, or electromyography, to track muscle activity. And this can be achieved with surface electrodes placed on top of the skin of the muscle, or via needles installed or needles inserted into the muscle fiber. And what EMG reveals is that movement is a highly coordinated activity, which involves muscle contraction at the same time with a relaxation of opposing muscles. To take an example, whenever we bend our arm, this is achieved by contracting the biceps and by relaxing the triceps. And conversely, whenever we want to extend our arm, this is achieved by contracting the triceps muscle and by relaxing the biceps. So whenever we talk about moving our arms to manipulate an object, or contracting the muscles of our legs to walk, fundamentally this is the mechanisms by which movement is facilitated. Okay, so then how exactly are muscles activated? What is the process by which they are allowed or they are told, so to say, to contract? Well, we do know from previous lectures that motor neurons play a fundamental role in that respect. Motor neurons, whether in the motor cortex, in the spine or the brainstem, are responsible for passing information through their axons all the way to the neuromuscular junction. This is the name of the specialized synapse that connects motor neurons to the motor end plate. And by motor end plate, I mean a folded postsynaptic membrane of the muscle fiber packed with neurotransmitter receptors. And the neurotransmitter in question is acetylcholine. This is what facilitates muscle contraction. It is released into the chemical synapse, which is the neuromuscular junction, and it binds to these receptors residing on top of the muscle end plate. Okay, so then you might imagine that an improper release of acetylcholine has quite visible impact on muscle functioning, and you would be correct. There's a high number of toxins and neurotoxins which can impact the release of acetylcholine. For instance, Botox, which is a popular commercial product, contains a small traces of a toxin that suppresses acetylcholine release, and in doing so, it relaxes the muscle and it provides a better surface appearance, let's say a less wrinkly one. In contrast, other toxins, such as a specific neurotoxin containing the black widow spider's venom, stimulates the release of acetylcholine release. And as a result of that, one may experience uncontrollable muscle activity or spasms. This is rarely a fatal condition. However, it can be quite painful, and it is treated by the use of specific antivenom. Okay, so this is what happens as a result of various toxins. What about of diseases or impairments in muscle function, more chronic conditions? Well, one of the conditions most often associated with an improper release of acetylcholine within the neuromuscular junction is called myasthenia gravis, or in free translation, grave muscle weakness. And this occurs because of postsynaptic membrane damage, and as a result of that, a decreased number of receptors on the motor end plate. This is a chronic condition. There is no cure for it. However, treatment can make life expectancy extend to the normal range, and also it can facilitate a lot of other activities, daily activities. I should also mention that nowadays there is quite a bit of research on trying to understand the impact of physical exercise on neuromuscular function and integrity. And there is quite a bit of work that points to the role of exercise in preserving or even improving function and structure at the level of the neuromuscular junction, and to combat the impact of aging. However, the results depend very much on what muscle we talk about, as well as by the type of exercise in question, whether endurance exercise or resistance training. Okay, so now if we go up one level into the spine, You might remember from previous lectures that this is where some motor neurons reside. And these are the neurons responsible for contracting, for sending signals to the muscles, and therefore responsible for their contraction. So also in the way of a review, you might remember that the spinal cord, has a rather straightforward division in terms of the organization of afferent signals traveling to the brain and efferent signals traveling towards the body and the muscles. That is, the ventral horns of the spinal cord are responsible for containing the efferent signals, and the dorsal horns of the spinal cord are responsible for containing the afferent signals. Let's try to illustrate that with a reflex movement. And perhaps one of the best types of stretch reflex is the patellar reflex, or the knee jerk reflex. And what we mean by that is a particular type of reflex which involves contracting a muscle in response to stretching the muscle. And that's exactly what a knee-jerk test essentially looks at. You are applying external force on a tendon of the knee, the patellar tendon, which results in stretching the quadriceps muscle, and then the information about this stretch is sent by the muscle spindle is communicated via the dorsal horn of the spinal cord onto a motor neuron, an alpha motor neuron. Hence, the motor neuron will react right away by triggering a contraction of the quadriceps muscle, protecting the integrity and the functionality of the muscle. So the muscle stretches, and then the alpha motor neuron tells the muscle to contract. Now, this is a simple enough mechanism. It is a involuntary movement, meaning that the motor cortex was not involved in it. Rather, it relies on a short chain of afferent and efferent signals, and that makes sense because reflexes are meant to be fast, they're meant to be efficient, and as a result of that, they should shorten that chain of receptors and motor neurons. However, for the most part in this course, we will be talking about voluntary movement. For the purpose of such a movement, as related, let's say, to the limbs or to the trunk, it is motor neurons, neurons of the motor cortex, which send their axons through the midbrain into the spinal tract. These are some of the longest axons in the human body. Some of them have lengths of over 1 meter. And it should be known by this point that they cross over on the other side of the body, so that they end up controlling the opposite side of the body. The more technical term for that is decussation. They cross over, and this takes place at the level of the medulla. Now, the main tract bundle of axons responsible for this is called the corticospinal tract, which is part of a pyramidal tract, and it bears that name because of its pyramidal shape when it passes through the midbrain. However, this is not the only tract responsible for motor control. So, for instance, another part of the pyramidal tract not shown here is the corticobulbar tract, which is responsible for the movement of the neck and the head and the face. In addition to the pyramidal tract, we also have extrapyramidal tracts. which rely on signals coming from subcortical structures such as the superior colliculus and the vestibular nucleus, and their function is not necessarily as prime movers, but rather as modulators. They're responsible for modulating posture or modulating speed when you walk and you run. So in that sense, they're also important, but they're not really the primary movers. One thing that is worth noticing here is the fact that they do not necessarily control the opposite side of the body. So while this may be the case for the tectospinal tract, which originates in the superior colliculus, we can also see here that the reticulospinal tract, originating in the reticular formation, controls the ipsilateral, the same side of the body, while the tract which originates in the vestibular nucleus is responsible for controlling both sides of the body. However, for the purpose of this course, we focus in particular on the corticospinal tract due to its main role and functionality. And again, it consists of signals sent by upper motor neurons of the motor cortex down the spine, where they synapse onto interneurons or lower motor neurons, such as alpha motor neurons, which in turn communicate the signals. They innervate usually multiple muscles throughout the body. In one of our previous lectures, we have also briefly touched upon what might happen if different parts of the corticospinal tract, or more generally the spine itself, bears damage. A intuitive way of visualizing that relies on the dermatome. Now, the dermatome refers primarily to the somatosensory system. In this case, the different parts of the body, of the skin, typically contiguous parts of the body, which send the signal to the same through the same nerve fibers. So, in case a particular part of the spine responsible for that area of the skin and of the body is damaged, then you expect that sensation is impaired or completely lost with respect to that part of the body. However, typically the damage to the spine is more diffuse, which means that not just the somatosensory signal is compromised, but also the motor signal, which involves the inability to control properly a certain part of the body. And in that context, we talked about different forms of plegia, paralysis of a body region, or paresis, which is a weakness or a partial paralysis of the body. So, for instance, if the damage occurs above T1, the T1 segment of the human spine, that would likely produce quadriplegia. That refers to the inability to move any one of the limbs, arms or legs. However, if the damage occurs below T1, let's say the level of T4, then this is a lot more likely to produce paraplegia, or inability to move the legs while the arms might be left intact in their functionality. And we talk about the limbs in this case, but the functionality of trunk movement might also be impaired or lost in both of these cases. Okay, so then what might happen if extensive damage occurs to the lower portion of the spine? Would that result in a complete inability to move our legs? And while you might expect that to be the case, surprisingly, this is not necessarily correct all the time. And we know a lot about this from animal studies. For instance, What happens if a resection, if a surgical cut through the spinal cord takes place in the spinal cord of a cat? Let's say at the level at which we expect that nerves to the hind legs would be completely damaged. And in a classical experiment, it was found that this type of resection at this level of the spine does not completely remove the ability of the cat to move the legs. So, for instance, if we place the body of the cat on a gurney to be able to support itself, and we place the legs of the cat on a moving treadmill, then what one might be surprised to see is that the hind legs still move to keep track of the movement of the treadmill. So again, the cat would not have the ability to control the legs enough to support its body. If we didn't have a gurney to provide support for the trunk of the cat's body, then the entire body would collapse, or rather the lower part of the body would collapse on the treadmill. However, if a gurney is provided and then the feet of the hind legs placed on the treadmill, then we would notice that the cat still has the ability to move the hind legs as a function of the speed of the treadmill. Is this a function of the sensory signals received through the feet from the treadmill? No, it is also found that in the absence of such signals, the cat can still move the legs. So then what's going on here? If you record muscle activity from the extensors and the flexors, you notice that this still occurs relatively normally. And we've been exposed to that in the context of our discussion about the biceps and the triceps and the recording of muscle activity in the human arm using EMG. So we do know that flexors, such as the biceps, which functions to bring the arm close to the body, and extensors, such as the triceps, which function to extend the arm, to push it out straight, work in opposition to each other. So one contracts while the other one relaxes. And the same happens in the case of locomotion. The extensors extend the hind limbs out, while the flexors bring them close in. So the extensors contract while the flexors relax, and vice versa. And this happens all the time. This is a rhythmic, stereotypical movement, something that we learn to do very quickly and efficiently. And it's not something that we need to think about or analyze very deeply. And the reason for that is because it relies on circuits of neurons called central patterns generators. And what this what these neural networks do is inhibit certain muscles while exciting others. So it's this complex pattern of inhibition and excitations that allows stereotypical common repetitive movements such as those involved in locomotion to take place. So in the context of locomotion, for instance, what an experiment such as that summarized here demonstrates is the fact that central pattern generators rely primarily on neural networks that do not include the brainstem or the brain, but rather rely on chains of neurons hosted by the lower part of the spine. So then a natural question that arises from this is what happens when damage occurs in humans? So for instance, if massive damage occurs in the lower part of the human spine, is movement still possible? Well, here the answer is a bit more complex, but what happens often, and we reason by analogy with what happened in the cat experiment here, is that the body of the patient is still placed in a gurney. Otherwise, a patient might not be able to support his or her own weight on the legs. But over time, the gurney allows more and more weight to be placed and supported by the legs. And then over time, bootstrapped by automatic reflexive movements, such as those supported by central patterns generators, what happens is through neural plasticity and through repair, self-repair of the spine and of the neural networks involved in locomotion, someone can regain partially or even completely the ability to move. Now, this can happen through extensive physical therapy, but sometimes that might not be sufficient. So over the last 10 years, we've seen some impressive novel types of surgery of the spine, which through the use of insertion of cortical neurons, for instance, neurons from the olfactory pathway into the spine, can help and can trigger repair of the neural circuits from the spine to facilitate regaining locomotory patterns and motor control over the lower limbs. So now let's see what happens if the impairment occurs not at the level of the spine, but that of the brain. And there are different possibilities for this impairment to occur. So a common occurrence is due to stroke. So let's say a stroke impacts this particular area of motor cortex. And if that happens, then in virtue of the topography of motor cortex with which we are already familiar, and its mapping onto different parts of the body, then we might expect that our ability to control the toes, the ankle, the knee, the hip, the trunk, and so pretty much the lower part of the body would be impaired. And we also know that the corticospinal tract, which originates in the motor cortex and then it dives down through the midbrain into the spine, decussates at the level of the medulla. So that means that the left motor cortex would control the right side of the body, and the right motor cortex would control the left side of the body. So the voluntary movement, which is controlled by the motor cortex on the left, in the left hemisphere, would, upon damage, impair or completely eliminate our ability to control the right part of the body. In this case, the right lower body. Okay, so then what might happen if an injury with the same size would take place not necessarily at the level of the cerebral cortex, but deeper within the white matter, at the location at which these fibers are all coming together into the corticospinal tract. Well, if that happens, you might imagine that the entire right-hand side of the body would be compromised, resulting either in hemiplegia, complete paralysis of half of the body, or in case the damage is partial and not complete, in weakness or partial paralysis of half of the body, in this case the right side of the body. So while generally speaking, the larger the area of brain which has impacted the injury, the worse the situation of a patient might be, the location matters as much. So injury with the same area more or less can result in symptoms and conditions of different gravity and different chances of recovery. Moving on. So far, our discussion has focused on pathways that link brain to muscles and facilitate voluntary movement. However, it's time to delve into a related but different topic. and that concerns the type of information that motor neurons contain. Presumably, whenever you're trying to engage in a voluntary movement, you have to encode a lot of information about where and how that movement occurs. And a fundamental aspect of that information is the direction of movement. So whether you want to move your arm directly in front of you or to the right or to the left, presumably there is some information at the level of the motor cortex with regard to that direction. And that is precisely what some of the most seminal studies, single-cell recordings in monkeys, established a while back. Intriguingly, it was found that motor neurons encode the direction of movement irrespective of the starting point or the end point of movement. So let's make that a bit more concrete. In the diagram shown here, we see a monkey moving the arm in one direction with respect to the center of a display, of a setting. So let's say that the monkey needs to move its right arm from the center of that setting in a multitude of directions. And then what we can do is measure the firing rate of that specific neuron in response to moving the arm in each of those directions. Away, to the right, to the left, towards the monkey, and so on and so forth. So the way in which we represent the firing rate of that neuron is by this diagram. So here each dot represents an action potential, and the more dots you see, the higher the firing rate of that specific neuron on multiple trials. Okay, so then let's see what happened with this specific neuron when the arm moves from the center of the setting towards the monkey. Well, and we see that indeed the firing rate seems to be maximized for these particular circumstances, when the arm moves towards the monkey or towards and slightly to the right, towards and slightly to the left. And that neuron is rather silent. The firing rate is minimal when the arm moves from the center of the display away from the monkey. So then what happens if we change a bit the experimental instructions, and the monkey now is to move the arm not from the center but rather from a different point, from a peripheral point towards the center of that setting. So let's say that the monkey moves the arm now from this point at 90 degrees towards the center of that setting. And interestingly enough, we see that the same neuron this time is highly active whenever the hand travels towards the monkey. So again, between the first experimental and the second experimental setting, what has changed is the start point and the end point of movement. But what remained the same is the direction of movement. The hand travels towards the monkey as opposed to away from the monkey. So based on this, we would conclude that this specific neuron is sensitive to the direction of movement and not to the specific location, whether starting point or end point of the movement. The other point that is quite relevant here is that the neuron is not sensitive just to one specific direction of movement, because we see the same neuron responding when the hand travels straight towards the monkey or towards the monkey and also to the right, towards the monkey and also to the left. So then how might we gain a better picture of what kind of directional information is contained in the firing rate of this neuron? And the specific plot that allows us to visualize that relationship is called a tuning curve. So what we can do here is measure the firing rate of a neuron across an entire range of directions. And in line with our previous observation, we see that there is one small range of directions that are explained or correlated with the highest firing rates of that neuron. However, there is only a small decrement in firing rate when the direction of movement changes a bit. And in reality, that a neuron is almost silent only when the direction of movement is orthogonal or in the completely opposite direction. So what we conclude based on that is that a neuron is not responsible just for a single direction, but rather for an entire range of direction, and there is a specific relationship that codes the contiguity of this directions in terms of related firing rates. So let's try to make that more specific with the aid of an example. Let's say that we are trying to move the arm mostly to the left, but also slightly up. Then you might expect that this is the result of a combination of different cells, one that fires vigorously to the left, and the other one which is responsible for moving the hand away from the subject, but in this case it fires only a bit. Then as a result of combining those two pieces of information with response to both direction and the firing rates of these neurons, we might end up with this particular direction. And if you're thinking about this in terms of linear algebra, for those who are familiar with this, you can think of each neuron providing a vector in space. Each vector, as you might remember from your math classes, contains information about direction and also has a magnitude. In this case, magnitude is provided by the firing rate of a neuron. So decoding the directional movement of the hand in this case is simply the result of vector addition or subtraction, for the simple example, just across two different cells. So in this case, all we're doing is just adding two vectors, one which strongly points us to the left and the other one that strongly points only weakly points the hand away from the subject. So this would be the resulting vector in this case. And to take the opposite example, let's say that cell 1 in this case has only a weak firing rate, but it is not silent, and cell 2 provides a maximal firing rate. So in this case, the result of adding the corresponding vectors triggers a movement mostly away from the subject, but also slightly left. Okay, so decoding the resulting direction of movement is simply the result of combining vectors corresponding to different cells. However, in the case of this simplified example, we are only looking at two cells. In reality, movement is encoded by large populations of neurons in the motor cortex, many of which trigger or specify the precise direction of movement, but some of which are also responsible for movements in neighboring directions. So for instance, whenever we're trying to move the hand to the right and away, then we have a large populations of neurons that respond to that specific direction. But we also have neurons that respond mostly to the away from the subject direction, and also some that are responsible mostly for rightward directions. And you can see how this population of neurons is sensitive to the different directions, and the combination of this entire population of neurons, or if you're talking about decoding the corresponding vectors, delivers the desired orientation of or direction of movement. So I hope this is made quite clear by this display here. But there is one other aspect of this display that I should draw your attention to. And this is the fact that we see activity in this neurons responsible for movement way before movement actually happens. So in the case shown here, shortly after a cue is presented to the monkey, let's say no longer than 100 milliseconds after a cue tells you in which direction to move the hand, we can see this neurons become quite active, and this activity can still be read out of the corresponding neurons for quite a longer duration. So if there's a delay period of 500 milliseconds, let's say, between the cue and a go signal that tells the monkey, okay, it's time to move the hand, we can still see activity within these neurons for this entire interval, which tells us that these neurons are involved in preparatory movement rather than just execution. So they prepare the body, they process information regarding the directionality of future movements. And it is often the case that we also see activity in these neurons during the actual execution, and that matches or mirrors the directional sensitivity that we find during the preparatory period looked into here during the delay. However, more recently it's been shown that the tuning curve of a neuron during the preparatory movement and during the execution of the movement might not necessarily be the same. And this triggered some very interesting investigations that showed that the information provided by a neuron is not simply about direction, but it is also quite context sensitive, and it also contains information about the force with which movement should take place, speed, and so on and so forth. And your textbook gets into a bit of detail regarding that. However, that doesn't change the fundamental point here that motor neurons do encode directional information. And this is certainly of theoretical interest because it provides us with a great deal of insight into how movement is generated and how it is coded at the level of the cortex, both in non-human primates and also in humans. However, this also has very interesting practical applications, and one of those practical applications is delivered by brain-computer interfaces aimed at re-establishing the ability to move. Imagine that the connection between the motor cortex and an arm is disrupted, let's say because of spinal damage. Then that connection can be established indirectly by relaying the signal read out from a neural population directly to the muscle. And there's a great deal of work nowadays, both in animals to study the efficiency and the likelihood of extending such BCIs to humans, but also plenty of work done in humans who have indeed suffered from injuries that prevent movement. So this is quite exciting, and one aspect of the of our discussion so far is quite relevant in the sense that we can read out the signals before the movement actually happens. So you can imagine that reading out this information a few hundred milliseconds prior to the time at which an artificial limb, for instance, is moved as a result of this readout does not seem disruptive too much to a human patient. So again, as a result of injury, motor activity or rather neural activity in motor cortex can remain intact, and reading out such information and relaying it indirectly to limbs can promote or facilitate, at least to some extent, movement in these individuals. Okay, now this might all be quite interesting here, but we're still left with a problem, and that is information about endpoint. While the motor cortex might be a reliable source of information regarding the direction of movement, what is responsible for encoding information regarding the endpoint of movement? And one thing that might come to mind is the segregation of visual pathways into a major division. You might remember that there is a ventral stream which is responsible for processing the identity and the category of objects. It facilitates visual object recognition, and there is also a dorsal pathway which is responsible for visually guided action. And it is the parietal cortex which is largely responsible for holding a 3D map of the environment. So it would be a good guess that the parietal cortex is responsible for providing information regarding the endpoint of a given movement. And if we dig a bit deeper into the functionality and the structure of parietal cortex, three regions are particularly relevant in this sense. For instance, one region, the so-called parietal ridge region, or PRR, is responsible, as the name suggests, for reaching towards an object, while a different region, the anterior intraparietal area, is responsible for grasping objects. For instance, adjusting the aperture of the hand so you can grasp an object correctly. Also, a relevant area is the lateral intraparietal area, or LIP, which is responsible for eye movements towards a given location in space, in your environment. So then you might assume that damaging or deactivating either or any one of these areas in isolation of the others might yield very different effects, and that expectation would be correct. So here is one example of a study that attempted to inactivate a part of the parietal ridge region in monkeys. In one version of the experiment, what happened is that the monkey was asked to hold a hand in a given position, and then a target was provided. The monkey was asked not to react, but to hold that position in memory for about 1 second, and then upon being presented with a go signal, the monkey had to move the hand at the previously presented location of the target. So the target is presented, you hold that location in memory for a bit, and then once the go signal is launched, then you have to move the hand to that particular location. Okay, so the question is, what happens if the PRR acts normally and whether it is inactivated? And what the authors of this study did was to measure the movement amplitude in that specific direction. So presumably the direction of movement is left intact, right? So the monkey would still be able to move the hand in the precise direction because direction is coded by the motor cortex and not by a parietal region. However, what you might expect to happen is that the amplitude of movement and hence the end location of the movement might be different, might be inaccurate when the PRR is inactivated. And this is exactly what happens. So if we look at the movement amplitude, before the PRR was inactivated and after, you see a significant difference in amplitude. That amplitude was shortened, hence the final location of the hand fell short of the actual location of the target post inactivation. And the authors also found that since not the entire PRR but a specific location of the PRR was inactivated, that difference between the amplitude of movement before and post inactivation was largest for endpoints that were presumably encoded by that by the neural population residing within the specific PRR subregion that was inactivated. What about saccades? What about eye movements toward that very same direction? The authors included a separate condition in their experiment in which the monkey had to hold the hand in the same position but just move its eyes towards a specific target. So the display here is very much the same, but the instruction is different. Instead of moving the hand, the monkey only needs to move its eyes to a given location. And in this case, again, we can measure the movement amplitude for eye movements rather than the hand. And the authors found no significant difference, if any difference at all, between the amplitude of the movement before and post inactivation. And this is presumably because the endpoint of the eye movements of the saccades is not encoded within the PRR, but rather in the LIP region. Okay, so then let's see what happens if instead of targeting the PRR, we target instead the anterior IPS, the intraparietal sulcus, which we believe is responsible for grasping. And as mentioned earlier, one aspect of grasping, which is very well documented, is the aperture of the hand. If I need to grasp an object for its midpoint, then presumably the distance between my thumb and my index finger is quite small. However, if I need to grasp an object across its longer axis, then the aperture, the distance between the thumb and the index finger needs to be modified accordingly. It will be quite a bit larger. Okay, so this is well known, and people have been tracking muscle activity in the fingers and the hand and the forearm to try to figure out the dynamics of such movements for a very long time. However, in one particular study, the authors included a rather clever manipulation, and that is they presented a stimulus, a simple object, that preserved its orientation or changed its orientation between the time human participant this time, initiated the movement, and the time in which the object was reached and grasped. So let's be a bit more clear about this. What happened is that you're told to grasp an object, and while you are reaching towards that object, the object starts to change orientation. So the object moves, and it can end up either in the same orientation that it started with, the one that precedes the initiation of movement, or it ends up in the opposite orientation. So where the beginning, in case it was horizontally placed, by the time you reach for it, the position of the object, its orientation has changed, so it's vertical. And we are very good at such tasks. We can correct the parameters of movement, whether by the arm or by the hand, in real time without much difficulty. However, what happens if you apply a disruptive TMS signal to the AIPS? And that is exactly what the authors of this study were interested to find. And what they did find is that there was difficulty in terms of modifying the aperture correctly in midair, so to say, when the TMS was applied at the level of the AIPS within 60 milliseconds of launching the movement. They also try to distinguish between a late and early delivery of TMS, but this matters a bit less. What matters is the fact that such difficulty and such inability to adjust the aperture correctly was not noticed when the TMS signal was applied to the motor cortex, to the parietal occipital complex, or to a different region of the IBS. A different way to visualize this is by looking at the time to reach peak aperture, and what they found was that this time was delayed in the perturbed condition relatively to the unperturbed condition. So what this tells us is that the AIPS plays a critical role in dynamic error detection during goal-dependent, visually guided, reach-to-grasp action. Okay, so now that we reached the midpoint of the lecture, let's move on to talk about a different region. And let's see what the human premotor cortex does. This is an area of the frontal lobe placed just in front, or rather anterior to the motor cortex. And you can see by its expanse, by its area, that it is quite substantial. This is shown in the rhesus monkey here in yellow, and we can see the same region portrayed here or colored in orange. So again, a rather large area of the frontal cortex placed anterior to the primary motor cortex. And researchers and neurophysiologists in particular have struggled to understand exactly what the function of the neurons in the PMC is for a while. A number of Italian researchers, led by Giacomo Rizzolatti at the end of the 80s and in the 90s in particular, provided some interesting observations that shaped our understanding regarding what PMC neurons do. What they did was, part by planning, part by accident, realized that some of the neurons in a PMC were responsible not just for movement. They weren't really firing only to, let's say, a monkey being involved in a manual task, but rather also when observing similar movement produced by others. So let's look at this examples of these diagrams from one of the early papers in the 90s. What the first diagram shown here at the top reveals is that one neuron was responsible both for firing when the experimenter, a human presumably, picked a piece of food, let's say a raisin from a table or from a tray, but then the same neuron was also responding quite vigorously when the monkey picked up from the tray the raisin. Okay, so the same neuron is responsible, or rather fires, both in observation of a movement or when the monkey itself performs the movement. However, if the human experimenter uses a tool to drop off the raisin on the tray, then no increase in the firing rate is observed in that neuron. Again, the monkey continues to respond at the level of that neuron with an increase in the firing rate whenever it picks up the food, irrespective on how it was dropped off on the tray. Also, interestingly enough, the same neuron fires vigorously whenever the monkey picks up that food in darkness. So there's no visual input, there's no visual guidance. Furthermore, if we look at a different type of neuron, instead of excitation, we see inhibition, in the sense that if the same human experimenter, let's say, picks up a raisin from the tray, then neuron responds by becoming relatively silent. And the same behavior is observed when it is the monkey that picks up that raisin. However, if a different type of action, let's say dropping off a piece of food on the tray is observed, then the neuron does not exhibit that kind of inhibition. of firing rate. So what does this suggest? Well, the hypothesis of Rizzolatti and colleagues was that in line with their observations, the same population of neurons appears to relate observation of meaningful actions and execution of those actions, and in that sense bridge gap, perform link between noticing the observations of others and being able to execute your own actions. So perhaps Rizzolatti and colleagues hypothesize that these neurons, which they called mirror neurons, are fundamental to understanding actions. And the way in which our brain tends to do that by relying upon such mirror neurons is by rerunning, so to speak, a scenario of the action observed as if it was an action performed by ourselves. Okay, so this is one of the earlier studies, but over the years there's been an increasing number of studies from the same group initially, trying to get a bit more detail on how exactly these mirror neurons function. And in a study published in Science in 2003, the same group led by Rizzolatti found that mirror neurons are responsible not just for observing actions performed by others, that is, when receiving both visual and auditory stimuli related to a meaningful action, such as, let's say, paper ripping or dropping a stick, but also simply to sounds, to auditory signals by themselves in the absence of visual input. So, for instance, if the monkey hears just the sound of paper ripping, in the case of this neuron, or just the sound of a dropping stick, in the case of this other neuron, then we can still notice a significant increase in the firing rate of those neurons. So let there be no confusion here. What we see at the bottom is just a trace of the auditory signal, while the main plots show the firing rate of the neurons, and these raster plots are simply convenient ways to illustrate the number of action potentials across a different number of trials, illustrated here as dots. So every action potential is a single dot in this raster plots. Okay, so now is it the case that these mirror neurons simply have selectivity for sounds, or rather sensitivity to sounds in general? Well, that doesn't seem to be the story, because there's no clear increase in firing rate if the monkey is presented just with white noise or with a series of clicks. Also, one might easily hypothesize that these neurons respond to meaningful sounds as related to some external stimulus. However, this is also not quite true, because if the monkey hears, let's say, another monkey call, so that might not necessarily correspond to an action, but it is certainly a socially relevant and meaningful signal, then the first neuron shown here does not exhibit an increasing firing rate, but rather an inhibition of firing. While in response to a monkey call, this other neuron shown here exhibits no apparent increase or decrease in the firing rate. Okay, so then if we try to look at an entire pool of neurons and verify their sensitivity to both visual and auditory stimuli, as long as they depict or represent meaningful actions, then we find that a majority of these mirror neurons are selective to both visual and auditory stimuli. And this provided further evidence in their eyes for the idea that mirror neurons facilitate action understanding, and in that sense that it transcends a particular perceptual modality. It can be vision, it can be audition, it can be tactile information. What matters is the fact that these mirror neurons bridge or link signals from different modality in the service of understanding action in others and in relating it to action as performed by ourselves. Now, let's see what other things are mirror neurons sensitive to. And here is another study from Rizzolatti and his colleagues at the end of the 2000s, published in Science, in which he investigated the sensitivity of these neurons to differences in peripersonal versus extrapersonal space. Peripersonal space refers to the closed space within which objects would fall into your reach, as opposed to extrapersonal space, a space which is further away from you and which is outside of your immediate reach. And what they found, interestingly enough, is that mirror neurons are indeed sensitive to this difference. So, for instance, cell number 1, shown here, seems to be responsible, or rather seems to react to movements, to meaningful actions performed within the extrapersonal space by somebody else, but it is no longer firing when the movement takes place within the peripersonal space of the monkey. So as expected, the neuron is firing vigorously when the monkey performs the corresponding action. So this is one cell. Let's look at another cell which has quite the opposite response pattern. It does not fire too much when the action takes place in the extrapersonal space, but it does fire quite a bit when the action takes place within the peripersonal space, as well as when the action is performed by the monkey itself. And let's take a look at the third type of mirror neuron, which acts vigorously when the action is performed by somebody else in the extrapersonal space, by somebody else within the peripersonal space, and also when the monkey itself performs the action. And if you look at about 100 neurons or so, what you find is that there's a bit of an even split between mirror neurons, which have sensitivity to actions both within the extrapersonal and peripersonal space, and the rest of them, which are more or less equally split between those that are sensitive only to actions in the extrapersonal space and only to actions within the peripersonal space. However, where things become more interesting is where sensitivity was measured in respect or with respect to the distance from the observer. And what this group found in 2009 is that there is sensitivity, as one might expect perhaps, for extrapersonal actions, so that this specific neuron, or rather this group of neurons, fires more vigorously the bigger the distance from the monkey. And in contrast, if we look at peripersonal mirror neurons, they tend to fire more vigorously the closer things happen with respect to the monkey. So the closer you perform a specific action to the monkey, the more vigorously this such mirror neurons act. Okay, but really the clever part of this experiment came when the experimenters decided to manipulate space, in the sense that peripersonal space can be transformed in extrapersonal space through the inclusion of a divider. And what I mean by that is that in the absence of a divider, we know what is extrapersonal and peripersonal space just based on the distance from the monkey. So let's take a look at an extrapersonal mirror neuron. It responds whenever action is outside the reach of the monkey as performed by the experimenter. But there is limited evidence for firing rate whenever the action is close to the monkey. So the interesting manipulation is when you place a divider so that the monkey can no longer reach to an object which otherwise would be placed within what we would deem to be peripersonal space. So effectively, by using a divider, we have converted the peripersonal space of the monkey into extrapersonal space. And upon doing so, despite the fact that the experimenter is grabbing this object in the proximity of the monkey, we observe that the same neuron suddenly becomes responsive to this action. So an extrapersonal space mirror neuron acting as a function of context. And we can also see, for instance, that other neurons, let's say peripersonal space mirror neurons, suddenly become inactive through the addition of a divider, which blocks the ability of the monkey to act upon the same object. So again, this is the result of converting what would normally be peripersonal space into extrapersonal space. Okay, so what does this show us? Well, it shows us that the response profile of mirror neurons are quite complex. There's a great deal of sensitivity not just to the intrinsic properties of a stimulus, but also there's a great deal of sensitivity on adaptability to context. And over the last 2 decades, the story surrounding mirror neurons became increasingly complex. For instance, in humans, it's been claimed that we also find mirror neurons within the primary motor cortex, within the parietal lobule, and even within the medial temporal cortex. So lots and lots of different cortical regions that appear to host variable numbers of such mirror neurons. With respect to the true function of mirror neurons, there's also quite a bit of a discussion in the field. Some people have claimed that they are foundational in explaining evolution, while some have been a lot less excited and speculative and claim that they are simply the result of some form of associative learning. So again, the hypothesis that Rizzolatti and colleagues initially put forward as a mechanism for action understanding is still one of the leading hypotheses in the field. Yet there are a lot of scientists who have challenged that hypothesis and have claimed that at best the theory behind mirror neuron system for action understanding is a simplified and incomplete story of the true functionality of this system. So there's still a great deal of research in the field trying to clarify and perhaps bring to an end a series of considerable dispute and controversy in neuroscience regarding the function and the relevance of mirror neurons in the context of cognition, both animal and human. Okay, moving on. The supplementary motor area, or SMA for short, is yet another region whose function has been extensively debated over the last few decades. This is a region located anterior to primary motor cortex and medial to the premotor cortex, much of which is actually located on the midline surface of each hemisphere. Okay, so what does it do? A number of studies have related the firing rates of neurons within the SMA with the performance of memory-guided actions, what we might be tempted to call automatic actions. So for instance, if we record from SMA neurons in a monkey, what we might find is that such neurons tend to fire a lot right before a pre-learned sequence is executed. So let's say that we train the monkey over and over again over a considerable number of repetitions and a significant amount of practice to turn a knob, then to pull it, and then to push it. So then what happens once that specific sequence of actions is well ingrained in monkey's memory, is that whenever the action is performed in this particular sequence, then we see a considerable pickup in firing rate of many of the SMA's neurons. However, if the monkey executes the set of actions in a different sequence, so let's say instead of turning, pulling, and pushing, the monkey pushes and then pulls and then turns, then we're not going to see the same result. Rather, instead of seeing a spike in firing rates before pushing, we see it right before the monkey turns the knob. Okay, so based on this, it is very tempting to conclude that SMA facilitates the execution of pre-learned automatic motor sequences. What about its role in the actual acquisition of those skills, in the memorization of novel sequences of movements? And a couple of years later, another group of Japanese researchers tried to answer that question. And they looked at two neighboring regions, one being the SMA, one being another region just anterior and neighboring the SMA. And this is intuitively called the pre-SMA, the pre-supplementary motor area. And what they found is that indeed, some neurons in the SMA were responding to the execution of rather to the learning of novel sequences more than to the execution of previously learned motor sequences. However, the majority of SMA neurons seem to be responsible for the execution of previously learned motor sequences. And this distribution, this relative proportion of neurons was reversed in the pre-SMA. The pre-SMA was primarily involved in the learning, in the trial and error learning of novel sequences, and fewer neurons within the pre-SMA were only or primarily responsible for the execution of learned sequences. And what was interesting about this study is that they didn't just look at this at one time point, but rather they assessed how the firing rate of different neurons changes over time in response to the acquisition of new motor skills. And it was interesting to see how some SMA neurons were relatively unresponsive while the monkey was learning a new sequence, but over time they became more and more responsive once the monkey became proficient at mastering and executing those movements. And the converse happened within the pre-SMA, where some neurons were very active during the learning stage, but then they became more and more quiet as the monkey successfully managed to keep track and to execute those specific series of movements. So this set of results is again well in line with the hypothesis that SMA, perhaps with some help from the pre-SMA, supports the execution of learned motor sequences. And there is convergent evidence from humans, not just from monkeys. And given that, you might expect that the SMA plays a critical role when you try to play a guitar or play a piano or even play rock, paper, scissors. But a lot of aspects of this research still remain to be clarified. So we do know that the SMA is involved in the initiation of voluntary movements. We do know that it has something to do with learned sequences, but it's yet unclear to what extent it is involved in things like postural control or manual coordination. And there are also quite a few discrepancies in the findings of animal researchers relative to people who've been looking at the function of DSMA in humans. And we're not going to have a lot of time to go into that today, but to conclude, there's plenty of work that still remains to be done to fully understand what role SMA plays in motor control and to disentangle between the many hypotheses that have been suggested in regard to its function. Last, we cannot have a discussion about action, about motor control, without bringing up the cerebellum. And it's been long known that the cerebellum is involved significantly in motor coordination. Now, because the cerebellum is such an old brain structure from an evolutionary perspective, it hasn't been paid as much attention as the rest of the brain. However, over the last 2 decades, it has become increasingly clear that the cerebellum is much more complex than previously thought, and it facilitates a few more, or rather a whole number of cognitive functions that previously we would have not imagined. However, since the goal of this lecture is to better our understanding of action and motor control, we will focus for a few minutes specifically on the contribution of the cerebellum to movement. And what we do know about the cerebellum is that it receives massive inputs, both from the sensory system and from the motor system. And while the anatomy of the cerebellum is quite complex, and these are by no means areas of the cerebellum or parts of the cerebellum that you have to acquaint yourselves with, I would only draw attention to the deep cerebellar nuclei, since they are the only output source of the cerebellum. So everything that comes from the cerebellum, whether directed towards the brain or to muscles in the body, goes through the deep cerebellar nuclei. Okay, so why is the cerebellum so important for fine motor control? Well, part of it has to do with its input and output. So, as I mentioned earlier, it receives massive input from the body, from our muscles. And it also receives, interestingly enough, information from the motor cortex. So, for instance, whenever the motor cortex sends signals to trigger voluntary movements to different parts of the body, to different muscles, it also sends a copy of this information to the cerebellum. So what the cerebellum appears to be doing is to build relationships between what the body experiences as a result of an action and what the commands sent by the motor cortex are that ultimately elicit those responses from the body. And I would point your attention to a subchapter in your textbook, which is specifically dedicated to what's called forward models. So this type of relationship between the expectancy of sensory consequences of actions and the actions themselves as dictated by the motor cortex. So what the cerebellum seems to be doing in this sense is to mediate, to provide an expectation. In response to a given action, what can we expect the sensory input to be? And in doing so, the cerebellum is not just able to facilitate fine motor control, but also to assist in learning, in motor learning. Okay, so these are some of the things that the cerebellum does. A lot of the information that we currently possess about the cerebellum, though, comes through studies of disease. So there is quite a few types of neurodegenerative diseases that impact the cerebellum. The cerebellum can also be damaged through stroke or through trauma. So a lot of these types of diseases or traumas results in what's called ataxia. And this is a very broad term that covers impairment of voluntary coordination of movement, often due to cerebellar lesion. So what happens in this case? There are different types of ataxia. For instance, dysmetria is a condition that involves poor or impaired judgment of distances. Whenever you're trying to reach for a specific object in space, you might have difficulty touching it or reaching it. You might be ending up undershooting or overshooting. You might be making quite a few mid-air corrections of the movement, and this is because of poor ability to coordinate between the visual input that specifies a distance and your ability to plan and execute movement that appropriately matches the perceived distance with the target of the movement. Another example of type of ataxia is dysarthria or slurred speech. And this should not be a surprise because being able to speak involved exquisitely well coordinated movements of the tongue, of the mouth, of the lips, of the vocal tract. So you might imagine that impairment of the cerebellum has an impact in your ability to speak properly. And perhaps many of us have had the opportunity to examine version or something akin to dysarthria as a result of let's say alcohol consumption. And this is because alcohol tends to inhibit activity within the cerebellum. So as a result of that, many fine motor control skills are impaired, and dysarthria is one of the most common effects. But so is dysmetria. So again, as a result of alcohol consumption, somebody might have difficulty reaching towards objects properly. And impaired gait or posture are also results of alcohol consumption, except that in such cases, these impairments disappear once somebody sobers up, while in the case of truly ataxic patients, let's say as a result of neurodegenerative diseases, this is not a condition that goes away, but rather it becomes progressively worse. So what are the problems that we tend to encounter or see when we look at people with difficulty walking or maintaining posture in ataxia? Well, this is just one example from a study that measured the parameters of movement while normal controls or patients would put on a treadmill. And what we see in these plots here are just contours of the fifth metatarsal. It's a small bone at the base of the little toe. And we see that the contours are more or less the same, superimposed at the same location on different trials. In contrast to what we see in normal, healthy adults, if we look at the same contours in patients, what we see first of all is that the distance between the 2 feet is larger. And this is something that they attempt to do in order to correct their balance. So they place their feet apart. They adopt a wider stance to compensate for their inability to properly maintain balance. The other thing that you see here is that both the precise position of this part of the foot and its orientation is quite variable from trial to trial. And again, this is the outcome of their inability to properly maintain their balance. And again, the precise level of impairment in ataxia is a function of how damaged cerebellum is, what is the extent, the gravity, and the specific location of the lesion. Now, it's a bit difficult to express or to provide a phenomenological experience of what any of these types of ataxia would manifest themselves like, but I've included a number of clips here to aid a bit that understanding. Okay, so there's a great deal that we know about the cerebellum, especially from the study of cerebellar lesions, but again, there's a whole lot more that we do not know, and that is precisely why this is such a fascinating part of the brain to study nowadays. Okay, so at this point, it's time to bring our discussion to a conclusion. And what we learned today is that motor control is a function of many different cortical and subcortical areas of the brain, whether in humans or in monkeys. And there is a division of labor across these many areas. However, there is also quite a bit of overlap in their ability to assist some known functions. So, for instance, we have learned that the supplementary motor area and the pre-supplementary motor area is valuable for learning and for executing sequences of movements. But we have also just learned that the cerebellum plays a key role in learning new patterns of movement. We have learned that the premotor cortex contains mirror neurons which might be involved in action understanding, but we have also learned that mirror neurons can be found also within the parietal cortex and possibly within the medial temporal lobe, and is part of a much broader network than previously thought that assists and plays a variety of roles for voluntary movement. We have learned that the motor cortex is responsible for the activation of muscles, and it is the corticospinal tract that is the main tract involved in motor control. However, we have also learned that the motor cortex is primarily responsible for voluntary movement, and there are a lot of other types of movement that fall under the umbrella of reflexive movement, for which much smaller chains or networks relying on alpha motor neurons in the spine might suffice. Okay, so movement is quite complicated. It comes in very different shapes and forms, and for this reason, we have an incredibly complex neural system that assists it, and it makes it possible. And this brings us to the end of the theoretical part of the lecture for today. So let's conclude with a brief discussion of the method section of journal articles. And I would start by noting that the method section is considered by many students as the most difficult part of a paper. Essentially comes down to explaining how the researchers did what they did. And it is critical to understand what's going on over there. However, this can be complicated because of the amount of jargon and technical knowledge involved. Yet I would encourage you to go through that method section carefully. Whenever I read a paper, which happens to be in my field of study as related to visual recognition, this is actually the part of a scientific article that I paid the most amount attention to. And whenever I read a paper which is outside of my immediate field of interest, I still go through the methods section quite carefully, irrespective of where it is placed. Right after the introduction, at the end of an article, or even in a supplementary online part of a publication. And the reason we pay attention to the method section so much is twofold. One is that it allows you to understand what is going on. So a lot of people make the mistake of just buying the arguments of a group of researchers as presented in the results section and the discussion, but without having a good understanding of how different things were measured and why, and going over all the details, you might miss some important problems with the study. A great number of papers in our field, in neuroscience, in cognitive neuroscience, have been challenged, and many findings have been contested and debunked due to flawed methods. So to some extent, you can think of the method section of a paper as the foundation. You might have an excellent writing of the results and some spectacular discussion presenting the interpretation of the findings. But if the foundation is flawed, then the results and the discussion do not really matter so much. And it is very much an exercise in futility. So understanding the methods is foundational in trying to critically understand the results, their validity, and also to assess the interpretation of the results in the context of broader literature. Another reason why the method section is so important is because if you happen to be a researcher in the same field, you might try to replicate that work, and without having access to all the exquisite level detail at the level of experimental design and analysis and so on and so forth, you might not be able to replicate that work. And replication is critical for scientific purposes. We rarely draw conclusions based on a single study. Rather, we seek convergence or divergence across a number of related studies. So if two different studies present you with different conclusions, one likely explanation of that fact could be that the methods are quite different from each other. So we can try to relate differences in results to differences in methods. However, if similar or identical methods yield different results, then we really have a problem that needs to be solved. So these are just two of the main reasons why the method section is so important. So then if you're a student in cognitive neuroscience and you don't have a lot of experience reading methods sections, then how would you be able to demonstrate or improve your understanding of this section of a paper? Well, first of all, I would emphasize the fact that you need a conceptual, a broad understanding of the experimental procedure. If you haven't been actively involved in research, and if you're new to this field of study, you're not expected to understand and to keep track of all the little details that an experienced, seasoned researchers might care for. So try to demonstrate a conceptual, a broad understanding of the experimental procedure, and keep track of key variables in play. So how would you go about doing this? Well, one thing that can be quite useful is setting up a diagram of the methods. So for instance, many of these methods section detail the number of participants and also their profile. Are we dealing with patients or with neurotypical adults? Are we dealing with children, with young adults, with older adults, and so on and so forth? Then we go into the things that are being measured, and it is often the case that a study presents multiple experiments, and sometimes the same experiment can be split into multiple sub-experiments. So what we've got here is a sequence of steps, right? So we can look, let's say, at a first experiment and ask, What did the authors of the study measure first? What are the relevant measures? Perhaps it's just a measurement of accuracy, or perhaps a brain scan relying on fMRI has been used. And then you moved on to the next experiment, and so on and so forth. And it is often the case that many papers in cognitive neuroscience present some version of this diagram to aid the understanding of the reader. But this has become less and less common in more recent years. So if such a diagram is present in the paper, great. If not, you might need to create one yourself. So let's look at how one of such diagrams might look like. So let's say we're looking at a typical fMRI study that involves about 20 healthy adult subjects, and then in the first part of the study, participants performed a picture judgment task that relied on that also was coupled with fMRI data collection. And let's say that participants were involved in two different tasks, one that involved shallow processing and the other one deep processing of the meaning of a picture. And then let's say that in a second step in this study, participants were asked to perform a memory task, which measured accuracy and reaction time and confidence, but no fMRI was collected here. And one presumably would be also interested next to relate the fMRI findings to the behavioral findings of the study. Okay, so now let's say that we're dealing with an even more complex study, this time with two different groups of participants. And let's say that we're looking at the impact that TMS-based disruption of neural processing might have at the level of a given cortical region. So let's say that we have 20 subjects who are intended to be placed within the TMS condition, and 20 control subjects who will not be administered TMS. And then we're going to proceed with all of these 40 participants to measure accuracy and reaction time in a reaching task. So let's say participants need to extend their hand to reach and touch an object. So this is what happens in the first stage. Then in the second stage, 20 of these participants will be performing the same task while TMS disrupts activity in a cortical region associated with motor control, let's say. While the other 20 subjects perform a second time the task without TMS. And this is important, right? Because some changes might be noticed from the first task to the second task, just in virtue of the fact that participants might be getting more experienced with the task, so there's a learning effect, or perhaps they're getting fatigued, and so on and so forth. So that's why it is important to have also a group of participants who do not undergo TMS. There's great value in having a control group here. Okay, so this is a TMS study that relies on two groups of participants. Then let's try to make things slightly more complicated. Let's say that we're looking at different types of patients. Let's say that we have frontal damage patients, we have patients with temporal damage, and of course we're also going to have a group of healthy controls, and ideally with like large and relatively equal numbers across groups. As it is often the case with patients of brain damage, we like to acquire from the very beginning structural data. So if we're interested in cortical damage, we certainly wish to acquire structural MRI data. If we believe that there might be some critical damage to white matter, then presumably we also want to collect some DTI. Okay, so with that in hand, we can proceed to the second step, which is collecting behavioral data from a battery of tests. So it's quite common, for instance, to conduct IQ testing and also measures of self-report function. How impaired do you think you are with respect to a given task? Let's say motor control, with respect to reaching, or let's say it's in the case of a visual recognition task, how impaired do you think you are with face recognition, and so on and so forth. And then we can proceed with the main part of the study. So let's say participants go through a first experiment that tests visual attention, to be more precise, sustained attention, while in a second experiment they go through a measurement of divided attention relying on multitasking. Okay, so this is how such a diagram might look for a relatively complex study that looks at the impact of different types of lesions on their impact with regard to visual attention. So what I'm going to ask you to do is take a look at the methods section of the following paper. This will be posted online. Again, your task will be to outline a diagram of the method section. And you can just proceed to reading the method section, or you can also skim through the abstract and the introduction if you so wish, to aid your understanding and to facilitate the processing of the method section. If you want, you can also look and go through a similar exercise using the paper posted last time with the corresponding lecture. Now, to make sure that you have a relatively good understanding of what's going on here, I'm also going to post my own solution to this problem, my own diagram, but I will delay that posting for a couple of days, and I would strongly encourage you to take a look and give this a shot prior to checking your result against the slide that I will post in a few days. And that is because often when you look at somebody else's solution, you might feel inclined to think that, yes, that's exactly what I would have been doing, but then you're missing an important aspect of learning, and also you might be mistaken. It is possible that in the absence of evaluating or looking at somebody else's solution, you might come across a rather different one, and there is value in that. So without further ado, good luck with the practice questions for next week, and I will be posting more detail about the second midterm by the end of this week.